For this video, we're going to focus in on mitosis and cytokinesis because this is an important part of the cell cycle that deserves a closer look, a little bit more information about what's going on. So if you remember from other videos, we can divide the cell cycle into two large groupings. We can call interphase all of the phases including G1, S, and G2, and then we have M-phase, which is where mitosis and cytokinesis occur. And an interphase cell, most of the time you see a cell, it's going to be an interphase cell. It's just going to look like a cell with a nucleus inside. So that's to give you a little bit of an anchor as we start thinking about what's going on during mitosis, is that when the cell stops looking like this, it's probably moving into M-phase, and it's doing some part of mitosis and cytokinesis. Now, just as a review of the cell cycle, there are a lot of things that need to happen right after a cell divides before it's ready to divide again. And these steps that occur prior to M-phase include the growing and making organelles, which occurs during the G1 phase. There's a G1 checkpoint, which is the part where the cell asks whether or not it grew and made organelles, whether it grew and made organelles or not. So once you get to the G1 checkpoint, then you go to S phase, where you need to duplicate DNA. That's an important process, and you'll understand why in a few minutes. Once you duplicate DNA, you go on to G2, where there's a G2 checkpoint to make sure that you've correctly duplicated all of your DNA, and you finalize a little bit of growing before you go into M-phase. So this is what's happening before M-phase, and now we'll focus in just on what's happening during the M-phase. So the M-phase can be divided into several different stages, and we're going to divide it into 5 different steps. And I'm going to talk about each step individually so that you can understand what's happening during them. So as the cell goes into mitosis, the first phase of mitosis it's going to be in is called prophase. And you can remember that because pro means before. Prophase is the first thing that happens. So during prophase, several things are occurring. The first thing that occurs is that all of the DNA that you saw in the previous slide in an interphase cell, which is all curled up like a pile of spaghetti, all of that DNA forms into chromosomes. And chromosomes are a way of taking that DNA and coiling it up really tightly, so it's in a nice, tight, compact package. And in fact, as we get ready to go into mitosis, you're going to have 2 copies of the complete genome of the organism. So each chromosome, which looks like an S, is going to be made up of 2 sister chromatids, 2 duplicate copies, which I've drawn here in green and red, of the same set of DNA. And the reason it's coiled up and tightly wound into a chromosome is because that will make it a lot easier when we need to divvy it up, spread it between 2 nuclei, so that you can pull it across the cell without it creating a lot of damage and getting tangled in a lot of things. So the first thing that occurs during prophase is the DNA forms into these chromosomes. The other thing that occurs during prophase is that the nuclear membrane starts to break down, and you can see here that the nuclear membrane is starting to have these little gaps in it. Now, it's easy to remember that the nuclear membrane needs to break down during prophase because all of the DNA is always found in the nucleus. If you start trying to move that DNA around, the first thing you need to do is get it out of that area, so you get rid of the nuclear membrane. The last thing that occurs during prophase, and it's something that may be more difficult to observe and occurs much later in prophase than the condensation of the DNA, is that microtubules, which I've drawn here in orange, will start forming at the MTOCs, the microtubule organizing centers, or we also call those centrosomes. So these microtubules are long strands of protein, almost like pieces of rope or poles that can reach out throughout the cell, and they'll become important later as they will allow us to grab these chromosomes and tug them to different areas. So this is prophase. Prophase, the DNA forms into chromosomes, the nuclear membrane starts to break down, and the microtubules are starting to form at these MTOCs. So once we've done all of these steps in prophase, we'll move on to the next stage. And the next stage is called metaphase. And so you'll see here that we've gone out of prophase. We no longer have a nuclear membrane when we get into metaphase, and these microtubules in orange have grown much longer. And what's actually occurring during metaphase is that the chromosomes are aligning in the middle of the cell here. They're lining up halfway between each of the MTOCs, each of the centrosomes, and they're forming what's called a metaphase plate, where these chromosomes are all lined up with one another. And you can almost tell from this picture the reason that they're lined up with each other is that our next step is going to be to pull them to opposite ends. But as you can imagine, it's critical at this moment that these all get lined up at the center of the cell. And this, in fact, is so critical that it becomes a checkpoint in the cell cycle. So I already mentioned that there was a G1 and a G2 checkpoint. There's also a metaphase checkpoint. And the metaphase checkpoint asks the cell, it pulls the cell, whether all of the chromosomes are lined up in the center. And if all of these chromosomes are not aligned in the center, the cell will not proceed until they are. Now, when you actually watch a video of cells going through mitosis, what you'll see is these chromosomes are moving up and down all the time. They're moving up and down, up and down, up and down. They're wiggling back and forth until they finally get aligned. And at that moment where they get aligned, then the next stage happens. So when we think of metaphase, what I want you to look for is I want you to look for chromosomes being aligned in the middle. And you'll see this nice microtubule structure leading up to these chromosomes in the middle. Now, once we've met this metaphase checkpoint, once we're past metaphase, we'll go to the next stage. And the next stage is called anaphase. And as you might expect, what occurs in anaphase is that the sister chromatids that make up those chromosomes separate. And you start using these microtubules to pull those chromatids to opposite ends of the cell. So this end, these MTOCs are pulling the chromatids this way. This MTOC is pulling the chromatids that way. And so by doing this, you're taking what was 2 copies of the genome, and now you're separating it into 2 discrete places. You're pulling those chromatids apart, and you're separating the cell into 2 poles. This is a pretty simple step, but when you see this, you'll see these chromatids moving away from each other at various levels. You can see them closer to the MTOCs. You can see them further from the MTOCs. But what differentiates anaphase from metaphase is that these chromatids have separated. They're no longer attached to one another. You no longer see X's representing duplicated chromosomes. You now see lines representing sister chromatids. Once the cell completes anaphase, it will proceed to its next step, which is known as telophase. Now in telophase, what you're seeing is that these sister chromatids are now decondensing, turning back into that pile of spaghetti that we talked about, and the nuclear membrane is reforming around these fragments of DNA. So when you get to the end of telophase, you've now divided your one nucleus that you started with into 2 nuclei. You have a nuclei here and a nuclei here. And just by virtue of the way mitosis functions, you're also going to have one MTOC, one centrosome in each end of the cell. And so at the end of telophase, you've really completed mitosis. You've gone through and taken one nucleus and turned it into 2 nuclei. There's one last step that needs to occur, though. And this last step is cytokinesis. And so when we look at cytokinesis, what we're actually seeing is cytokinesis is the cutting in half of the cell itself. So in cytokinesis, these 2 nuclei are still where we saw them before, but the cell membrane will start to pinch in. And this cell membrane will pinch in so that it divides this into 2 individual cells. And once you've done this, you've completed cytokinesis and you now have 2 unique cells. So the cell is splitting into 2 cells during cytokinesis, and we call these 2 cells the daughter cell. We start with a mother cell, and we end up with 2 daughter cells. And once you go through cytokinesis, the cell cycle is going to resume at the G1 phase. So these guys are going to start growing again. Once they've grown large enough, they're going to duplicate their DNA again. They're going to grow a little bit more, and they're going to re-enter mitosis all over again. Now, one note about cytokinesis is that in plants only, because plants have a cell wall, you have the formation of what we call a cell plate. And this cell plate forms during cytokinesis to really form a wall between the 2 cells as they're also pinching them off into individual cells. And you can see this in this drawing. Here's a cell at the end of telophase. Once it's at the end of telophase, you start getting cell wall pieces dragged into the middle. And these will form a wall between the 2 cells, and then the membrane will break off between the 2. It's the same idea as cytokinesis in an animal cell, but it's a slightly different step that you have this cell plate here. So just to review one last time, all of the components that go into mitosis and cytokinesis. You first have prophase, which is the step where the DNA condenses, the nuclear membrane breaks down, and you start forming microtubules in the cell. Once you complete prophase, you then go to metaphase. Metaphase is the stage where the chromosomes are going to align on the metaphase plate. They're going to line up in one place in the center of the cell. That's a checkpoint. As long as everything is aligned, it'll continue on to anaphase. Anaphase is the step where those chromatids will be pulled apart to opposite ends or opposite poles of the cell. After anaphase, you then go to telophase, where these chromatids will decondense. They'll turn back into a pile of spaghetti DNA. The nuclear membranes will reform around. And that's the completion of mitosis, and then you'll follow it up very shortly after with cytokinesis, which is where the cell is actually going to divide into two cells. So prophase, metaphase, anaphase, telophase, followed by cytokinesis. If you remember all that, then you will be really comfortable with talking about mitosis.